Well, thank you, Matt. Boy, it's interesting to hear yourself introduced. In any case, thank you very much for inviting me to give a talk today and for presenting the work from my lab group and my collaborators group. And as you can see here, we'll be talking about the Medicago genome, the Medicago truncatula genome, as a tool for looking at complex gene families that are involved in symbiosis. In the course of today's talk, what I hope to be able to tell you about a little bit, tiny bit about modulation, but more looking at the story of the complexity of gene families that are involved in plant microbe interactions. A little bit about the process of genome sequencing and the discovery of variants, structural variants, and the challenges of trying to understand, to discover and characterize structural variants when you're using sequence data as a basis. Okay, rhizobial legume interaction, nodulation is a critical point. Just want to say that I have to, oops. Symbiosis is central for nitrogen fixation, something that I'm sure everybody in this room knows about. And Medicago truncatula is a model legume for looking at that, for asking that question. There's a nice picture of Medicago and something that you've probably seen in the hallway because it's something that the University of Minnesota has really taken a lead in doing the analysis of. Now, when we talk about symbiosis, it involves a complex set of interactions and communications. And just as a reminder, just to remind everybody, so it involves, first of all, rhizobial bacteria that live in the soil, the root, and the root hair, which is really the site where the interaction takes place. What happens is those roots are, the rhizobial are engulfed in and then eventually induce the plant to form a root hair, and that corresponds, that sort of coordinates with the induction of cortical tissue, which proliferates and eventually forms the nodule, where the bacteria fill that nodule and lead to the production of fixed nitrogen. And you've probably heard about a lot of the molecular pathways that have been involved in nodulation, because this is a very high profile, very profound breakthrough in the field of plant microbe interactions in the last 25 years. They involve factors like things you might have heard of called NFP, DOMI1, DOMI2. So these are, in some cases, they're receptor kinases, they're calcium transporters that are involved in moving calcium, or also they're molecules that are involved in transferring the calcium message from the membrane to the nucleus. And in the process, we've learned that mycorrhizal interactions, the interaction of mycorrhizal fungi in the soil, and symbiotic interactions with rhizobium share a pathway, and so many of these factors are shared between these two different kinds of symbiotic systems. Now, in my lab and in the broader Medicago group here we have at Minnesota, we're interested in large gene families that play a role in plant microbe interactions and in resistance and symbiosis. And here I'm just showing a phylogenetic tree which highlights the coiled NBS LRRs. So this is a very large gene family that frequently plays an important role in disease resistance, in reaction to plant pathogens. Some of these genes are also known to play a role in symbiosis. So we're interested in large gene families, but we're especially interested in a gene family called the nodule-specific cysteine-rich peptides, the NCRs. And here I'm just showing a multiple sequence alignment of those NCRs to give you an idea that there are hundreds of them, they're complex, and they've got a lot going on. And the characteristics of these NCRs, well, first of all, one of the things that I need to tell you is that they have a signal peptide. So they're molecules that are secreted extracellularly. They have this very regular patterning of cysteine residues, which is very highly conserved among different NCRs. And that it's a large family. There are 800 of them in Medicago, and they're only expressed in nodules. I should also say that this family of nodule-specific peptides, they're also closely related to defensins. So the defensin superfamily is already involved in plant microbe interactions, and here in the case of Medicago, there are 800 of them that are involved in the nodulation process. Now, working in the lab, Deanna Trujillo is a graduate student in the plant biology PBS program. She's been looking at small secreted peptides. She's been looking at nodule-specific peptides. She's been asking the question, where are they located? What different plant taxa do you find them? And one of the important observations that she and others have made, so she's expanding on work that's been known for 10 years, is that they are primarily found in Medicago and its close relatives. The name for this taxonomic unit is the IRLC, the inverted repeat lacking clades. So this group includes Medicago, alfalfa, and trifoli, the clovers. So those are the primary places that you find it. But I should say that in August of this year, so 2 months ago, it was discovered that another taxon within the legumes that is not in the same branch as Medicago, called the Dalgibaceae, also have these same NCRs. So it appears to be an example of these defensin-like molecules being sort of co-opted for use in nodules by multiple legume lineages. So now what is it that NCRs are supposed to do? Well, I took a recent review and I sort of blew up one of their pictures and tried to make it kind of a good illustration of what it is that people think NCRs are doing. So here, NCRs are known to control bacterial terminal differentiation, and it works in a funny way. So here's the infection thread where bacteria are sort of being introduced into the root of a plant, and this is supposed to be a picture of a plant cell. And what's happening is that in the case, NCRs are these small peptide molecules which have these conserved cysteine residues, and they're involved in plant microbe communication. They're already known to be involved in plant microbe communication. Well, when you have an appropriate interaction, when you have a rhizobium of the correct strain, a strain that is the right kind of a strain that the host recognizes as an appropriate partner for nodulation, that those NCRs, first of all, they promote its differentiation, which is terminal, so that the bacteria can no longer divide, and they change their morphology and they become optimal for doing the nitrogen fixation process. And in cases where the bacteria has mutations, which is shown down here, where it's not able to interact with it's not able to produce transporters that can move peptides across the membrane, or if it either has that mutation or it's an inappropriate strain, a strain that's not recognized as a nitrogen fixing partner, what those same NCR molecules do is they lead to the death of the bacteria. So either the peptides, and there are 800 of them, remember, they're either making a differentiated bacteria that's suitable for doing nitrogen fixation, or they're actually acting like an antibiotic, like a toxin against the bacteria. Well, I first gave this talk, the talk I'm doing now, or a very close version of it, about a month ago, about 6 weeks ago, in Amsterdam. And so I just, I happened to have a couple of extra days while I was there, and I went to the botanical garden in Amsterdam. I don't know if you've ever, I don't know if anyone else has had a chance to go there. I thought, oh, there you go. And it's a beautiful place. It's extremely well laid out. One of the fascinating things about it is it has this series of concentric ring plantings, which have the whole angiosperm taxonomy or diversity laid out in it. So it's really cool. Well, while I was walking, of course I'm in the Asteraceae, and I'm working my way down to the Magnolia, and I'm going on all these different different plant groups. I get to the legume group, and I run into a plant that looks like this. And this looks familiar to me. And so you can see that, I don't know if you remember, but these look just like Medicago leaves. The only thing is, it's in a giant plant. So this is like about maybe 6 or 8 feet tall, wide, 3 or 4 feet tall. It's as big as this table here. And so I was fascinated by it, and it made me think that, the roots of this plant, there'd be a lot of roots. There's a lot of roots going on there. And those roots have got a lot of NCRs inside of them. Like kilogram quantities of NCRs, because it's very highly expressed. By the way, this is called Ononis ripens, which is also called rest harrow. And that particular member of the trifoli was such an important weed that it was called a rest harrow because it tore up the farm equipment that people used trying to get rid of it. So I thought that was a good story. So that I left that slide in the talk. Okay, so because we're interested in plant microbe interactions, we're interested in legume rhizobial interactions, and we're using Medicago as a tool for doing that. One of the first steps we took was to resequence the genome of Medicago Originally just 20 or 30 times, and since then almost 300 different times. And to give you an idea of where those samples came from, Medicago is a pan-Mediterranean species. It grows primarily on the coast. The collecting sites can be exciting and fantastic. In fact, these ones over here in Israel, last year I went with Brian Stephenson on his trip to Israel, and I did some collecting of Medicago samples just to supplement some of the taxa or some of the lineages we didn't have covered as well. I actually didn't get the right lineages, but now we have some new Medicagos that weren't stopped at customs. And then this gives you an idea about the diversity of the kinds of morphologies that you might see among different Medicagos that are going on here. Well, the important point of this slide is that what we were doing was we were taking 200, 300 different Medicago isolates, like these guys, and we're extracting them and getting them ready for sequencing, and then sequencing them to either a low or a medium depth so we can look at the diversity between different Medicago strains, between Medicago isolates. And the reason, frequently when people do this kind of analysis, the goal of that is called genome-wide association mapping. And I'm not going to spend too much time on GWAS. That would be a story of its own, and that's not going to be the focus of today's talk. But in the GWAS, the question you're asking is, okay, I'm looking at the genomes of 300 different Medicagos. I'm not just looking at the genomes, I'm actually looking at all of the variants that exist inside those genomes. And the variants are things like single nucleotide polymorphisms, Snips, and there are deletions and translocations and inversions and so on. So we can take the sequence of those 300 individuals and align them together and look to see where the nucleotides differ and where they're the same. Where we find those differences, we ask the question, are the nucleotide differences, are the SNPs, or the translocations, or the inversions, are any of them correlated with some sort of phenotype of interest? And to us, of course, the phenotype of interest is nodulation. So we sequenced these 300 Medicagos and did this analysis where we're looking at all of the SNPs in the Medicago genome, and SNPs being, remember, single nucleotide polymorphisms, and then correlating them with nodule number or nodule efficiency. Okay? And so that's what a picture like this shows. Here are the 8 chromosomes of Medicago, and there are 2 million dots on that screen. Each one of those 2 million dots is one of the nucleotide polymorphisms that exist among the 300 accessions we sequenced. And we're looking for correlations between whether there's a polymorphism, and in this case, how many nodules are in the top 5 centimorgans of the root. And so you can see that there are some that go up to a very high significance, like 10 to the minus 7, 10 to the minus 8. So the first step of this project was simply to look for the SNPs that have an association with nodulation, and then that was the starting point for the analysis. Okay, so Sean, who's sitting in the audience here, is a person who actually went through, found candidate genes, and is trying to analyze them in one way or another. The first thing we discovered was that many of the hits that we find were genes that people had already known were involved in nodulation, just like you'd expect. So remember I told you in a slide about 10 slides ago that there was a gene called NFP. So NFP, sure enough, is one of the hits that we get. And I told you that calcium signaling plays an important role in nodulation. And sure enough, a calmodulin that had previously been associated with nodulation came up as a hit. But also interesting, a number of different novel candidates came up as being significant. In fact, even more significant in some cases than some of the historically known ones. So we wanted to analyze those. And one of the first ones that we did was this ubiquitin conjugating enzyme. And what Sean's been doing, working with Roxanne and other people in the lab, what they did was they actually knocked out we've taken a number of different ways to knock out the function of this candidate gene. Here it is right here, candidate gene. It's on chromosome 4. The chances that this happened by chance was 5 times 10 to the 6, so highly significant. And we're looking at a mutant population where we actually knocked out the gene by having a transposon jump inside of it. This is a collaboration with the Nobel Foundation, which is the group that developed that mutant population. And the question is, okay, if a line that has a mutant that's jumped into that ubiquitin conjugating enzyme, does it have is the rate of nodulation affected? And that's what you're seeing over here, so that wild type versus mutant, that you see that the number of nodules that are formed in the root, either the entire root or a 5-centimeter section, is dramatically affected by it. And that actually there's something of a difference between different strains of different rhizobial strains. Well, very recently, Sean just this last week, along with a cluster of people who are working together, we're looking at a number of the other candidates. I'm not going to go through the details of all this, but you get the idea that, and I should say that I'm not showing you the candidates that failed. So there are some candidates that we thought something was going on and there was no statistically significant difference. But here, for example, here in this DNA heat shock protein, you can see that there's a dramatic effect on nodulation at the top in the first 5 centimeters. And you can see it's like less than 0.00001. And I'm showing you just a number of cases where either nodulation is reduced in the mutant, or in this case, where this homeodomain leucine zipper knockout actually increases. And there are different ways to knock out. I told you about the transposon, but some of these, let's see, these right down here, these are using this very cool technique called CRISPRs, where Sean and a variety of groups over in Cargill are using I'll almost call it a prepackaged. It's a very standardized method to either induce local mutants or to cause deletions that can eliminate the function of a gene of interest and then ask whether there's a phenotypic effect. So we can either do it with transposon populations, or we can do it with CRISPR or other related gene editing techniques. Okay, so that's a cool story, isn't it? That we were able to use this population level resequencing technology to identify genes that might be interested, because Snips, nucleotide polymorphisms of those genes, are showing statistically significant correlations with the trait. Then we knock out the trait, we knock out the gene, and we affect the trait. But we're actually more in the lab, we're equally, or even more so, interested in structural variation. So not just individual base pairs that are changed, but big changes in the structure of the genome. We're interested in insertions and deletions and translocations and regions of the genome that are in one individual and not even in another. So something that is often called pangenomics. So we're interested in exploring that part of the gene space and trying to understand the relationship of, or the role of structural variants and novel gene segments in nodulation and interactions with microbes, especially symbiotic microbes. So I want to tell you a little story about Joseph Gulen, who's a graduate student in Peter Tiffin's lab. And what he's doing is he's using a kind of a very quick and efficient way for looking at something called copy number variants. So one line, remember we have 300 lines, one line might have one copy of a gene, another one might have 3 or 5 or 10 copies of that same gene. And that's a kind of variation that is potentially very important as well. And sure enough, the top hit that he found, this one that I'm sort of highlighting here, it is a copy number variant of NCRs, the gene family I told you before. So we hypothesized that variation in NCRs or NCR gene numbers or NCR gene family architecture plays a role in symbiosis, because we know there are proven examples where the NCR proteins interact directly with the rhizobium and they control the efficiency or the success of nodulation. But it turns out that our ability to discover structural variants is not very good. I mean, ours, the field, I mean, everybody, not just the Medicago group. Turns out that discovering structural variants is very difficult. And I just took a quick shot from a Nature Genetics review to show you what I mean. So here you can see there's a genome, and there's some other. There are 2 genomes: a reference genome and one of the 300 ones we were sort of resequencing. And we find, and imagine that there really is a deletion there. But now all of the reads, all of the sequencing reads that we use when we do Illumina, the next generation sequencing technique that's so common right now, if we do that, all of our reads are really tiny short guys. And so the only way we're going to be able to see whether there's a deletion is if one of those little tiny reads gets cut in the middle. And then we're going to have relatively short segments on either side, because you probably know that an Illumina read is only 100 bases. So if you've got your deletion and it overlaps it, you've got like 25 on this side and 65 on that side. And then you're trying to say, was there really a deletion? Is that really the way it is? Could we got them up, flipped around, or could it be hitting somewhere else in the genome? Or what's going on? And here's just a thing where it's an insertion, where actually here you find that the pairs of reads don't line up the way they're supposed to. So this turns out to be a relative. It does not have the kind of power that anyone would want for making these kinds of discoveries. So the big effort in the last 2 years the big effort, in addition to the knockouts I told you about a few minutes ago, was we wanted to actually do full genome resequencing and use that as a basis of looking for structural variation. So that's really been, along with the functional analysis that I was telling you about with the knockouts, it's the resequencing of genomes and using that as a basis of structural variant discovery that I want to talk about. And so, let's see. Oops, no, got ahead of myself. So what I would like to say is that our group and collaborators at a number of institutions, who I will recognize at the end, I apologize, I haven't shown them yet, we have actually undertaken the resequencing of 25 different Medicago genomes. And by this, I mean not just sort of like putting them in a next generation Illumina sequencer and letting the thing sort of spit out a shotgun sequence, but actually assembling them all de novo and having high quality assemblies that we can then compare among them and also to the reference. And one of the first things that we did was actually say, okay, let's look at all of and this picture is just a phylogenetic tree showing you the different Medicagos that we've resequenced, including this very special one called HM101 or A17. That's the reference. And that reference is a very high quality so-called backbased reference. So it's nearly of the same quality as what you might find for Arabidopsis or rice. Not quite there, but nearly so. And these other ones also are having very high quality assemblies that we can compare to one another and to A17. Well, one of the first things we did was we asked, well, are those assemblies, does the overall assembly make sense? And one way to do that, we compared genome size based on fluorometry, sort of like actually staining nuclei and asking how much DNA is there, and the sizes of the assemblies that we created for those. And we don't think anybody has actually published this data before. We think data like this before. So it's actually kind of cool. It turns out that there's a remarkably good correlation between the genome sizes predicted by and these kind of assemblies, by the way, they're called all paths. So it's all paths LG is a very reliable and very high quality way of assembling genomes. And so the all path assembly and the genome size are very highly correlated. What is this? Oh, you can't read it. It says 0.84. So there's the R for this is 0.84. So a very strong correlation between fluorometric estimates and the genome assemblies. And then in work that was led by Peng Zhao, who's a graduate student who just finished here, I think, early in the summer. He actually led the assemblies and sort of the analysis of the assemblies. And I'll tell you a little bit about how to interpret genome assembly data. So in this column here, you see something called Scaffold N50, and then you see a list of, I think, 16 or 17 different Medicagos that we analyzed here. And you can see that the numbers here range from about 300,000 to 2 million. That's something called a Scaffold N50. And what that's saying is that the chunk of DNA sequence, the chunks of DNA sequence that we were working with, that we can describe 50% of any given Medicago genome sequence in chunks that are 500,000 base pairs in size. So we're not looking at little pieces that are 100 bases. We're looking at pieces that are 500,000 bases. So there are still gaps, and there's still a lot of, they could be even better than they are. But the questions we can ask are on the scale of 500,000 base pairs, where before we were looking at a scale of a few hundred bases. So it's a much more powerful way to be asking these kinds of questions about structural variation. And sure enough, so we use those high quality assemblies, we align them to one another into the reference of Medicago, And then we asked, well, how many structural variants do we find? Because this would have been a relatively hard question to ask if we only had early next generation sequence data available to us. What you can see is that this HM056 is the most closely related to A17, to our reference. And this HM022, in fact, this cluster right down here, those are the ones most distant. Taxonomically, we know that to be the case. And you can see that anywhere from 10% to more than 30% of the genome is involved in some kind of structural variation. So think about that for a moment. Do you remember how many Snips did I tell you was on that GWAS? I said there was a couple of million on that analysis. And in fact, if you look at the entire gene space of Medicago, there are about 5 million common Snips in the Medicago genus. Well, here, 34%, so what is this? The sum of all of this is 10, 40, so there's 115 million base pairs in this HM022 that are involved in some structural variation. A deletion, an insertion, an inversion, a copy number change, something like that. So that much more of the gene space that's variable between us and also between Medicagos is involved in structural variants, not single site mutation variation. And I told you just a minute ago that in the case of Medicago, structural variants in the NCR gene family control nodulation. Because I showed you a case where a copy number variant in an NCR had this enormous, well, it had a really significant effect on nodulation. And so what do these variants actually look like, if you actually look at what they look like? So now this is a piece of DNA. Let's see, what's the scale here? There's a kilobase. So this looks like about 20 kilobases that's being compared to something else where it's about 30 kilobases. So so something that's 20 kilobases long is big enough to fit in like 4 or 5 genes. So there's space there for 4 or 5 genes. We're comparing it to another thing that might have 5 or 6 genes. And what we're finding is that by lining these up, where everything's lined up, there's no gaps or anything like that, there's a missing piece there. You can see that that's a much more powerful way to look for insertions and deletions because you're actually looking at the deletion or the insertion there. Here's a case where there's an insertion. By the way, it's HM101 is the reference. So we're finding a deletion in HM101, an insertion in HM101, an inversion, a translocation that involved kind of a flipping of the sequence. And each case, well, this case down here, that's 5 kb. So this is close to 50,000 base pairs. So 50,000 base pairs is a relatively large piece of DNA that we're talking about. And that's where all of these numbers come from. Short indels, large indels, copy number variants, and so on. Now, another thing that we could do, because we have these complete assemblies, we could ask, well, what about the genes we're interested in? Well, we're interested in transposons. A while ago, I told you we're interested in NBSLRRs, and we're also interested in the NCRs. And the NCRs, by the way, are a member of a larger gene family called the cysteine-rich peptides. Remember, they had cysteines at specific intervals, and that cysteine-rich peptide family is an important family to look at. In fact, we actually, Pong, as part of his thesis, actually wrote a pipeline, an informatic pipeline, to look for CRPs, for cysteine-rich peptides and other small peptides like that. And so you can see, one of the things you can see is that all the way from all these different Medicago genomes, that, for example, the number of NBSLRs doesn't change at all, or changes very little. So that it goes from 700 to 860. So the number, at least within the Medicago truncatula species, the number of NBSLRs doesn't change very much. And that's quite significant to a plant path audience, because those NBSLRs, they represent probably 80 or 90% of the resistance genes that you all are playing with. Whether you're in a greenhouse or whether you're sequencing, NBSLRs play a major role in plant pathology. And the number of genes is staying relatively constant. And the same thing for CRPs. And about half of these CRPs are actually this NCR family. So the numbers stay quite constant, so that was an interesting discovery. We could look at the distribution. We could look at, with high resolution, we could look at the distribution of different gene families along the chromosome. And so this x-axis is chromosome 5. And then on this top one, the y-axis is the density of NBSLRRs or CRPs. So you can see, for example, there's big spikes of NBSLRR density those are clusters of NBSLRs that are living right next door to one another. And sure enough, they're correlated with regions of the genome that have higher levels of variation, of both base pair and structural variation, as you'd expect. So when we have de novo sequence from so many different genomes, we can actually make a curve that, what we would call a novel sequence frequency spectrum. This is actually a pretty cool thing, so let me describe what it is in some detail. So what you're seeing right here, it's 1, 2, 3, all the way up to 15, because we're only looking at 16 genomes. All these different colors that are squeezed up in the 1 zone, that's the size, that's a representation of the genome sequence that's specific, that's only found. It's not found in any of the other 16 accessions. So this little blue one here, and this little purple one here, and this little red one here, and that green one, and even that different color purple, that represents the amount of sequence in each of the 16 genomes that's only found in that one genome, not found in any of the other 16. It's a private piece of DNA. Just like all of us, we've got some private DNA that no other human being has. We don't have that much, but we've got some. Well, in the case of this Medicago comparison, these range from about 1 million base pairs up to, what does it say, up to about 7 million base pairs. So there's a few million base pairs of completely private DNA. And then the rest of this curve is how much of that DNA that's shared among different accessions. And so there's, for example, there's another 1 million base pairs that's shared among 14 out of 16 of the accessions we analyzed. Okay? So we hypothesized. Well, in fact, I'm going to show you some cool things about. So remember that this kind of DNA is something we're able to pull out of our analysis and look at. And this kind of frequency spectrum is a basis for looking at what's called the pangenome. If you've never heard of the pangenome, it's basically a statement that's saying, well, my DNA doesn't cover all the humans, and sequencing just A17 of Medicago doesn't cover all of the Medicagos. So, but when we sequence 16 of them, we're capturing a pretty good representation, especially if they were well selected, a good representation of Medicago gene space. So that's what's indicated here, and that gene space can be described in two different ways. The top curve, which is starting to level out but clearly has not saturated, is about 450 million base pairs. So in sequencing the 16 accessions, if we look at all the DNA that is in the reference, plus new stuff we found, the second genome we looked at, plus still newer stuff we found when we looked at the third one and the fourth one and the 16th one, starts to level off at about 450 million base pairs. And of that, 270 million base pairs is found in all 16 of them. And that's called the core genome. So this is the core genome, is the 270 everybody's got. And the difference between these two, that's called the dispensable genome. Because obviously you can still be a Medicago and not have it, because there is at least one Medicago that doesn't have that chunk of DNA. So that's like a pangenomic analysis. Now there are two important things there are many important things, but there are two important things that we found from doing these analysis I'm showing you here. The first one is that Snips, remember Snips? I used to talk about Snips, now we're all in structural variation. But we made a cool discovery about Snips. Snips are profoundly underestimated with read mapping approaches. So when all you're doing is next generation sequencing, and you're sort of aligning it to your reference, and you're saying, is there a base pair substitution here? Is there a base pair substitution? You do it across all of the genome. When you do that, you underestimate, you under-discover, you fail to discover as many as 70% of the SNPs that are actually there. So this is a big number, like 70%. And it turns out we're not the first or the only people to discover this. In fact, it was first discovered in humans, where they have spent more money and were doing this kind of high-quality de novo sequencing earlier. But so just to give you a graphic image of that. So if you look at the blue region, that turns out to be the chunk of DNA that both our high-quality analysis and the old, fast and dirty next-gen of analysis found. But this purple here, those are all new SNPs that would have never been discovered if you only did next-gen sequencing and you didn't build assemblies and actually do this where you did alignment level, synteny level comparisons. The other thing that we found that's quite important is that the families that are most frequent in the novel gene space, remember this space here, we're talking about this space here, the genes that are most frequent in that gene space, what are they? The NBSLRRs and the NCRs. So the gene families that I'm talking about, they represent most of the new gene space. There's also a lot of transposable elements because there's just a lot of transposable elements. And so you're always going to find new transposable elements in the novel gene space. But among gene families that have PFAM domains that are definable, the ones that are most common in the novel gene space are the ones that are involved in plant microbe interactions. In fact, I would actually make an even stronger statement than that. I would say that the most important genomic difference between any two individuals, or any two plant individuals, is the array of plant microbe interaction proteins that they have in their genome. That's the thing that's still varying. That everybody, when you've done your 100th Medicago accession, there'll still be some novel new genes, and there'll be NBSLRRs or NCRs. Okay, well, it turns out that these two gene families that I've been talking about have very different variant architectures. So I'm going to take the last couple of slides to try and illustrate that for you. So here, NBSLRRs exhibit a high level of rearrangement. Insertions into the gene, deletions from the gene, or rearrangements among the gene. And I'm just showing you, I think, there's some highlighting here, an example of what I'm talking about here. So this and this, these two red circles are kind of capturing an NBSLRR gene. Remember, there are 750 of them, but this is just one of them. And what we find is there's a remarkably high level of rearrangement. See how what's happened is it's lost some of its exons. So the gene has been deleted, and some of those exons are gone. And presumably, that means we've got an NBSLRR that has a different specificity. It interacts with a different avirulence protein, or it interacts with a different plant protein in a surveillance type setting. But in other words, we can find, and I can show you lots of examples of where orthologous genes are rearranged with respect to one another. But that's not what we find when we look at NCRs. We don't find NCRs being rearranged and domains shifting around and so on and so forth. Instead, what we find are lots of copy number variants. So here, there's 2 going to 1, 4 going to 2, looks like 8 going to 10, and this one I think is 3 that goes up to 20 copies. And by the way, you would never be able to see this with short read, unassembled alignments. That would just be impossible. So in other words, both of these gene families are big, they're clustered, they're involved in plant microbe interactions, they're involved in nodulation, and yet the way they differ from one another, the way variation is structured in the population is very different. Rearrangements on the one hand, a higher frequency of rearrangements in the case of NBSLRRs, a higher frequency of copy number variants in the case of the NCRs. And here's another way to illustrate it. What I'm showing at the top is, this is one subfamily of the auxin-inducible domain family. Without going to lots of, it's a gene family, and there's a bunch of them in the genome. So it's a little bit like NCRs and NBSLRs. But it's kind of a boring, we would consider it a boring gene family. It's certainly not involved in plant microbe interactions. Here's a subfamily of the NBSLRRs, and here's a subfamily of the NCRs. So these subfamilies have like 30 or 50 members in them. So to give you an idea of what we're looking at. What I'd like you to see is that in the case of the NBSLRRs, this little rectangle is showing you the novel sequences that are there. Remember the novel sequence that I was telling you about a few minutes ago? Well, you can see that for almost all of the NBSLRRs, it's one only. Only one of the accessions has that particular NBSLRR. That one copy, that one representative of it is a rearrangement that nobody else has. It's a private rearrangement that an NBSLRR has. Whereas when you look at the NCRs, which you see is a very different thing. Yes, it's in the novel gene space. These are novel genes that weren't found in A17 or something else. But the way it got to be a novel one was you took a gene that was there and you made 3 copies of it. Or you took a gene that was there and you made 5 copies of it. And so you got 4 new copies of a gene. They're novel. They weren't in the original sequence. But rather than rearranging it, you just make copies of it. Now remember that the total copy of NCRs stays the same, so we're also losing NCRs as much as gaining them. Well, I've been kind of bragging about the assemblies we've made and how cool they are, but the fact is that even if you use the all-paths assembly and you have 100x coverage and you really spent a lot of time making good assemblies, you're still not going to be doing as well as you ought to. And so a group in the lab, and it involves, let's see, Jason Miller, Joanne Mudge, well, Jason Miller, who's at JCVI in Maryland, Joanne Mudge, who's a former graduate student, and she's a senior scientist at the National Center for Genome Resources, and Kevin Silverstein, who's at MSI. They've worked together to develop a PacBio, which has even longer sequence reads as a way of making even better assemblies and discovering structural variants that even we're missing even with our de novo assemblies. And I'm just going to show you one example of that. So this is an all-path assembly, and this is a PacBio-based, which they call alpaca-based. And in all-paths, all they find is a deletion. Something's different. When they do the all-paths, what they find is there's 12 copies of an NCR. And that NCR is actually, well, I shouldn't call it an NCR. It's a CRP, because that's a CRP that's found in mycorrhizal interactions, not in nodule interactions. So different plant microbe interaction. Okay, well, that's the whole story I have. So what did I tell you today? Well, what I told you was that, in fact, I'm going to read it from here because I got this microphone. What I told you is that NCRs, the nodule-specific cysteine-rich peptides, are taxon-specific. They're only found in Medicago and its close relatives, but they have a dramatic effect on rhizobial development. That we can use GWAS, genome-wide association mapping, that technique of looking at millions of SNPs and then looking for correlations with phenotype. We can use GWAS to discover novel nodulation candidates, including copy number variants among the NCRs, which is a reason why NCRs sound so interesting to us. I mentioned that read mapping, so the older technique, the one from long ago, like 2012, 2011, the old method of finding SNPs underestimates them by 70%, and that underestimate is even worse when we try to use old next-gen read mapping techniques to do structural variants. That's what SV means, structural variants. That NCRs and NBSLRs are the most variable of all the different kind of gene families, and they're highly overrepresented in the novel or dispensable genome space. So when we look at those genes, we find lots of genes and lots of transposons. The ones that seem most variable are the ones I've been talking about. NCRs and NBSLRs have notably different variant architecture. In the case of NBSLRs, they are overrepresented for rearrangements within the gene, whereas in the case of the NCRs, they're overrepresented for copy number variants. And finally, I make the case, I hope it's clear by now, that in order to study structural variation altogether, you really need to make these high quality assemblies. You can't use short read technology. And in fact, so it'd be advice to you to be skeptical of anyone who gives a talk and all they're talking about is using short reads to look at structural variants. That would be a very inefficient way that would miss a lot of important questions. A lot of people were involved in this. I recognized graduate students and postdocs from my group that were involved, but we're just part of a consortium that has a dozen different centers involved. And I especially want to recognize the co-PIs that are here at the University of Minnesota. Let's see, Mike Sadowski, Kevin Silverstein, Bob Stupar, and Peter Tiffin. And then our collaborators at other institutions. Joanne, I mentioned at NCGR. Betsy Martinez-Vaz, who's at Hamlin. She's worked closely with Mike Sadowski's lab. She's also led an initiative we have in kind of undergraduate education for underrepresented minorities. Maria Harrison, who's at Boyce Thompson at Cornell, and then Jason Miller, who's at JCVI, which used to be known as it's the J. Craig Venter Institute. Let's see, so is there anything else I wanted to say? Well, I hope I've made the case that, first of all, that structural variants are interesting. They're hard to look at, but they're especially important in the case of plant microbe interactions and the families that are involved in plant microbe interactions. And I would predict that even though many of you don't do genome sequencing, that underneath the phenotypes you're looking at, the gene families that I described today, and the variants, the kinds of structural genomic rearrangements that I described, that's actually that's the underlying genetic cause for the things that we as a department like this should be interested in. Okay, so with that, I'll close, and I'm happy to answer any questions you have.